Hello, everyone. The topic of my presentation is dynamic evolution of unstable genomes. We know that cancer is known to be the disease of the genome, and genetic alterations underlie tumorigenesis and resistance to therapeutics. Also, genome instability is known as the hallmark of cancer. In this presentation, I want to share with you some of our recent findings about evolutionary dynamics of unstable cancer genomes and how this process might contribute to tumor development. Before discussing cancer genome evolution, I want to briefly revisit the classical model of tumorigenesis known as clonal expansion. As cells replicates its DNA and divides into daughter cells, errors can occur, resulting in mutations or alterations in the genetic sequence that can be stably transmitted into the progeny of the cell. It has also become clear that in addition to alterations in the DNA sequence, alterations such as DNA methylation and chromatin states can also lead to a result in changes in gene transcription that can be stably transmitted into the progeny population as well. Now, if either by genetic alterations or epigenetic alterations, the cell acquires some proliferative advantage, it can lead to clonal expansion, upon which further mutations can arise and lead to more aggressive phenotypes. This was the first model of tumorigenesis proposed by Peter Noble in 1976, originally proposed for the development of leukemia. Since then, it has been adopted in many other contexts, and most perhaps best known in the context of the development of colorectal cancer, known as devogogram, where the different phenotypes and genetic alterations are associated with disease of colorectal cancer at different stages from early to late. When we think about cancer evolution, the process is not much different from a general evolution process, where we think of the cancer cells at different stages to contain and or harbor different sets of genetic mutations that give rise to the different phenotypes, including the potential to invade surrounding tissues and metastasize to distal organs. It is conceivable that even for cells with the same genetic makeup, that is, they have the same genetic material, there could still be substantial variation in the cell phenotypes, including the cell types. For example, early stage disease could tend to be associated with more differentiated cells, whereas more malignant, aggressive, and high stage disease tend to be associated with poorly differentiated cells. Now, in this presentation, I'm not going to talk extensively about this phenotype-genotype relationship in the context of tumor evolution. It's a fascinating question. Instead, my focus will be on the genomic evolution. In other words, we try to understand the process where the cells acquire different genetic alterations in the course of tumor development. In trying to understand genome evolution from the perspective of mutations, we want to understand two questions in particular. The first question is, what is the spectrum of genetic diversity? In other words, we try to understand what kind of genetic alterations can arise in the progeny population, even if all of the progeny cells started from a single ancestor cell. On top of that, we are also interested in understanding how all the alterations arise. In other words, what is a possible trajectory from a single ancestor cell into one of the terminal state? It is helpful to make some distinctions about different types of genomic alterations that can arise. One of them I call local variants, which include both simple base substitutions, as shown here as an A to T change, as well as insertion deletions of one or multiple nucleotides. In contrast to local variants, another group of variants are known as structural alterations or structural variants. Structural variants do not change individual nucleotides, rather they change or alter the organization of large chunks of DNA, including generating deletions or insertions of large chunks of DNA, kilobases or megabases long, generating duplications, inversions, or transpositions, and finally, but also very frequently in cancer, leading to translocations, sometimes between different chromosomes. To give you a snapshot of the complexity of structural alterations that can appear in cancer genomes, I'm showing you 4 non-representative examples of cancer genomes sampled from the literature. On the very top left corner, you can see one cancer genome essentially consisting of a single set of every chromosome, whereas next to it on the right, there is a cancer genome that's almost hexaploid. In other words, contains about 6 copies of every individual chromosome. And we also see intermediate states where you have near diploid state or near triploid state. In addition to the numerical aberrations in the number of chromosomes, there's also extensive structural abnormalities in the composition of individual chromosomes, shown as the individual chromosomes making up with different colors. Despite such diversity, what we know is that all of these different cancer genomes must have arisen from the same ancestral state that is essentially a diploid human genome that makes up all of the cells in a human body. So my lab is interested in the question of how or what mutational processes are happening or driving this process when the single ancestor cell with the diploid genome content could evolve into this very divergent terminal state. Of course, in trying to answer this question, we are not really interested in just recapitulating the individual steps of alterations that make this transition, but rather we try to understand the underlying mutational processes and the biological mechanisms. Why are we interested in this? I guess besides intellectual curiosity, one observation connects this process to the clinical relevance, which is we often see structural alterations that arise in the same time when the disease transits from low-grade or more benign disease into high-grade or invasive or metastatic disease. Exactly how the generation of chromosomal alterations can be related or contribute to the transition from pre-cancer to cancer is unknown, but we think that by understanding how these alterations arise, we can learn or gain insight, new insight into this transition, and which may help us find better biomarkers to predict disease progression, as well as uncover new vulnerabilities associated with high-grade disease. So my presentation today will focus on the evolution of cancer genomes, and in particular, the evolution of structural alterations in the cancer genome. Before we talk about our work on this understanding the mechanism, let's first review what are the causes that can lead to genome instability. There has been a lot of work in cell biology and molecular biology generating insight on what causes genome instability. People have already attributed causes such as defective DNA repair, including defective error correction during DNA replication, the generation of replication stress, disruption of DNA damage response, defective spindle assembly checkpoint, which can lead to chromosome missegregation during cell division, telomere crisis, and cell division errors. How exactly can this one or multiple mechanisms listed here contribute to genome instability is still not completely understood. Now, in my presentation today, I actually want to talk about one simple mechanism that could contribute to genome instability, which is unstable chromosomes. Unstable chromosomes can result from one or multiple mechanisms, including some of the causes listed on the previous slide. Two common paths leading to unstable chromosomes are shown here. The top one is known as telomere attrition. We know that a linear chromosome consists of a centromere, shown as the red dot in the middle, and the two telomeres, which protect the naked ends on the chromosome. As cells divide, the telomeres get shorter and shorter over each division, and when the telomeres get critically short, it can be deprotected and expose a naked DNA end. This is known as and the naked DNA end is unstable because of all the DNA repair pathways that would try to join naked DNA end. When the telomeres gets critically short, cells can undergo a catastrophe known as telomere crisis. Another simpler mechanism that can generate unstable chromosomes is reciprocal translocation. Think about a scenario where two double-strand DNA breaks were generated on two different chromosomes, and the two chromosomes underwent an exchange of genetic material known as the reciprocal translocation. Now, there are two possible ways that the two double-strand breaks can be joined between the two chromosomes. Shown on top is the outcome when the two breaks are joined in a way such that the resulting translocated chromosomes are still stable, meaning each chromosome has a centromere with two telomeres, except that the partners have swapped. Now, there's another possibility, which means that which generates the outcome that the two terminal segments are joined and the two centric or two chromosomal pieces with centromeres are joined together. In this case, the reciprocal translocation creates an acentric fragment and a dicentric chromosome. As will be shown later, the dicentric chromosome is unstable and can lead to the generation of two broken chromatids during cell division. The evolution of broken chromatids was perhaps the first form of genome evolution being observed in eukaryotes. In fact, it was being observed before we even know what chromosome was made of. Barbara McClintock first made the observation that the generation of broken chromatids can lead to the formation of anaphase bridges during cell division, and the bridges can persist. The generation of bridges which be broken during cell division can lead to the formation of a new bridge in subsequent mitosis. This creates a pattern known as breakage-fusion-bridge cycles. This slide summarizes the different steps of a breakage-fusion-bridge cycle. The first step is the generation of a dicentric chromosome. You can see the two centromeres the two chromosomes with centromeres are connected at the end. And mitosis, the two centromeres are being pulled to opposite side of the spindle and to different daughter cells. At some point, the chromatid bridge, the chromosome bridge which connects the two daughter cells will be broken, resulting in two broken chromatids in each individual daughter cell. The broken chromatid can be replicated. Now, the broken end is replicated as well. The broken ends can then join with the broken end in the replicated sister chromatid, creating a new dicentric chromosome out of the fusion. This is known as the fusion event. Now, the new dicentric chromosome can undergo a new round of bridge formation and breakage event. And the cycle can keep going, which makes it breakage-fusion-bridge cycles. As you can see, the first in the zeroth generation, when dicentric chromosome was formed, nothing really happens. By generation one, the two daughter cells would have different genetic makeup. One daughter cell had a loss of the terminal segment, and the other daughter cell had a gain of the terminal segment. Each round of breakage-fusion-bridge cycle would create two daughter cells with different genotypes, one with a gain of the terminal fragment, the other with a loss. And you can see that marked by number 1 here, one daughter cell can acquire terminal gains over consecutive cycles. And the outcome of that is gene amplification. Shown on this slide is an example of ERB2 amplification, which is a frequent oncogenic event in breast cancer. In this case, it has been inferred that the ERB2 amplicon follows the pattern of a breakage-fusion-bridge cycle. Now, this is the terminal stage of the genome, and we do not really know for sure how exactly this amplification arose. And the topic of my presentation today, I would like to, we are trying to combine in vitro experimental system with single cell sequencing to try to gain more insight into how this process happens, and including how the oncogenic amplifications can be generated. So the general framework of analysis is the following. We started off from a cell where we can engineer this abnormality. In this case, the abnormality is a dicentric chromosomal bridge connecting the two daughter cells. As the two daughter cells progress through the cell cycle, the bridge gets broken. And then as the cells, we can either harvest the cells to sequence them to understand the genetic and mutational outcome of bridge resolution, as well as we can let the cells grow further into more progeny populations, and then do the sequencing there to figure out the long-term evolution consequences of bridge resolution. I should mention that this work is a combination of two experimental and analytical approaches. On the left, it's done primarily by live cell imaging, and on the right is done with single cell sequencing. And this is carried out by a bunch of talented postdoctoral fellows and research scientists shown here. And this is a project that we work closely with David Pellman's lab in Dana-Farber Cancer Institute. So let's first talk about how we generate chromosomal bridges or broken chromatids. The most specific way of generating a broken chromatid is to introduce a site-specific DNA double-strand break. In this case, we have introduced the double-strand break using CRISPR-guided Cas9 activity. By generating breaks when the cell was in the G2 cell cycle phase, we can create two breaks on the sister chromatids. There's a certain chance the two sister chromatids can fuse at the break ends rather than joining the terminal segment, creating a dicentric chromosome. The dicentric chromosome would not be sensed until the cell undergoes mitosis, and when the two centromeres are being pulled into opposite daughter cells. We watch for the resolution of the dicentric bridges, essentially abscission of the dicentric bridge between the two daughter cells. And then we take or harvest the two daughter cells or the progeny of these two daughter cells, and then do single cell or bulk whole genome sequencing to understand the genetic outcome. So let's look at what happens in the first generation. That is, we start with a cell and let the cell divide into two daughter cells connected by a chromosomal bridge, and wait for the bridge to resolve, and then sequence these two daughter cells separately. Schematically, this is shown here in the dicentric chromosomal bridge. We don't expect the break to be exactly symmetric between the two centromeres. If the break is not happening exactly in the middle of the chromosome, you would imagine that one daughter cell would inherit a broken chromatid with slightly less material, whereas the other daughter cell would inherit the broken chromatid with more material. In other words, one daughter cell has a terminal loss, and the other daughter cell has a reciprocal terminal gain. And indeed, this is what we see from the single cell sequencing data. Shown here are the real sequencing data of, in this case, the one and only chromosome that we inferred to be the bridge chromosome. And the gray dots show the DNA copy number of the particular allele that was being affected, or the particular chromatid that was in the bridge. And you can see that in daughter 1A, there is a terminal DNA loss. The copy number goes from 1 to 0. That is accompanied by a reciprocal gain in daughter 1B at the same segment, exactly as we had expected. So that is simple enough. In one generation, we see terminal gain and terminal loss. What happens in the second generation? So we went on and let the cells divide again, in this case, giving rise to 4 grandchildren. Of course, we won't be able to take their, we take the cells or the mother cells of these granddaughters for sequencing because we let the cells divide. Nonetheless, we can easily infer the kindred relationships between these 4 cells. You can see that the top 2 cells, granddaughter cells, both have terminal gains, whereas the bottom 2 cells each has a terminal loss. Looking more closely, you would see the middle 2 cells share a break end, just as we see in the 2-cell stage, and we infer the breakpoint to be the red line, shown as the red line, which is exactly where the reciprocal gain and loss between the middle 2 cells happen. Now we notice that if you look at the top cell and the bottom cell, the gain segment in the top cell and the loss segment in the bottom cell both span a larger genomic territory. And the explanation for this observation is that the bridge chromatin is not fully replicated. Shown here is a schematic view of this explanation. So the 2 sister chromatid underwent the first cell division. There's part of the chromatid was in the bridge. This part of the bridge was not replicated, even though the rest of the 2 chromatids are both replicated completely. Now, as the bridge was resolved to generate into 2 granddaughter cells from each side, this middle part of the bridge chromatin was not replicated and is only inherited by 1 granddaughter from each side, whereas the other 2 granddaughters do not inherit any piece of the bridge chromatin. And that explains the pattern of loss of DNA material in the bridge because of lack of DNA replication. Again, we learned that the breakage-fusion-bridge cycle is not exactly as it was described in the original model, because the lack of DNA replication of the bridge chromatin implies that over many generations, chromosomes in the bridge would be gradually lost over the population. We took one step further. In this case, we grow the daughter cell, which inherited a broken chromatid, into a clonal population. We want to look at what happens in this clonal population. Shown here is the bulk average DNA copy number of one such clonal population where we have engineered a broken chromatid on chromosome 4. And you can see the DNA copy number of 2 different homologs of chromosome 4, shown here in red and blue. In particular, we recognize that a lot of genetic alterations or copy number alterations here are obvious, evident on the 4p arm. And we think that was because the break was engineered at the telomeric side of the chromosome, and that resulted in the generation of these alterations, both on the red and the blue homologs. Now, the generation of the break was not allele specific. Therefore, we are not surprised that the broken chromatid could actually be generated on both homologs and resulting in the accumulation of alterations in both of them. Now, one feature I want to emphasize is that if you look at the DNA copy number of the blue or the red homolog, normally for one cell with a single or both homologs, you would expect to see an integer DNA copy number of either the red or the blue allele. Here, you don't see integer copy number. In fact, the DNA copy number is almost always between integer states. And our interpretation for that, instead of the cell having non-integer number of molecules, it rather implies that the population of cells consists of different clonal populations, and the different clonal populations might have different copy number states of this different, both the red and the blue homologs. So we decided to dig deeper into this. To resolve this, we took individual cells from the progeny population, grow these individual cells into a clone, and performed, again, bulk whole genome sequencing to look at a DNA copy number. The results are schematically shown on the right. The blue bar represents the presence of the blue homolog, whereas the absence of the blue reflects the loss of the blue homologue in the population on top. In the bottom, we also see that the blue homologue is overwhelmingly lost from every individual cell that has been sequenced. Now, what makes this even more interesting was that the red homologue underwent segmental gains, but different patterns of segmental gains in the population above and the population below, separated by the solid black line. All of the cells on top share the same pattern of blue homologue in terms of DNA copy number, whereas the cells in the bottom panel showed varying copy number states of the red homologue. Most importantly, the breakpoints or the segmental breakpoints of the copy number changes in the top population and the bottom population are totally distinct. We can perform heuristic clustering of the individual cells based on the copy number states, and shown here is the heuristic phylogeny. Because of the scarcity, due to the scarcity of copy number events, we would not be entirely confident about the progeny, about the phylogeny of the different cells, but we can recognize at least there are two main branches. The top branch showing this all share this red copy number states drawn in a rectangle, dashed rectangle, and the bottom population show different copy number states of the red homologue. To show you how we made an inference from the real data, this slide 22 shows the original sequencing data of DNA copy number of the red homologue and the blue homologue, with the schematic view of the segmental copy number states drawn below the sequencing data. As you can see that all of the different cells share a common ancestral breakpoint in the red homologue on the telomeric side, very close to the telomere, but individual cells follow different evolutionary trajectories and acquire very different patterns of segmental gains of the red homologue. I've marked the private breakpoints, segmental breakpoints, using green vertical lines, and we occasionally can see shared breakpoints between different single cell subclones, shown here as the magenta vertical line. One mechanism that we can gain further insight into the evolutionary trajectory is to use the pattern of chromosomal rearrangements that we detect in the different subclones. The chromosomal rearrangement tells us how the different gain segments are being joined together. In the interest of time, I'm not going to go to the detail of how we use the rearrangement pattern to infer the phylogeny, but simply showing you the result of our reconstruction of the different events in the course of evolution. For example, the first event marks the initial ancestral break on the telomeric side that is accompanied by an internal translocation followed by a translocation to chromosome 14. After that event, the two branches started off doing on different evolutionary trajectories and acquire different alterations. I want to emphasize that even though we only have information about the genotypes in the progeny cells in the terminal state, by looking at the relationship between these progeny cells and constructing a phylogeny tree, that allows us to infer the trajectory of evolution, shown here as the dashed arrows. In order to gain further resolution into the amount and degree of genetic diversity in the population, we further performed single cell profiling. In this case, we selected hundreds of cells from the progeny population and profiled their DNA copy number using a recent assay developed by 10x Genomics, which allows us to interrogate DNA copy number of hundreds of single cells in a parallel fashion. Shown here is the DNA copy number heat map of 500 cells of chromosome 4. So each row here corresponds to the DNA copy number at 1 megabase bins on chromosome 4. In the next slide, we zoom into the region of the P arm, where we see the most genetic variation and diversity between the different copy number states. And I highlighted the different copy number states we have previously detected in the subclonal population and their correspondence in the single cell profiling. Again, you can see the consistency between the selective sampling from single cell subcloning, which would only allow us to sample a limited number of individual cells genomes, and the single cell profiling, which tremendously expands the resolution into a cell-to-cell variability in the genotype. Now, this is one sample that already gives us a lot of insight into the amount of genetic variation that can arise from a single ancestral break. In the next example, I'm going to show you a sample where the evolution takes a different turn. Again, let's start from the population level, the bulk average DNA copy number in the population of cells. Shown here on top is the average DNA copy number of the red and the blue homolog, and the blow up of the P arm is shown in the bottom. If you look at the red allele, you can see there's a lot of dots that oscillate between copy number 1 and approximately copy number 2, essentially reflecting segmental amplifications or segmental loss. Whereas if you look at the blue allele, it shows largely a loss with an average population copy number between 0 and 1. Let's first look at the blue allele. Again, we take single cells from the progeny population, grow them into subclones, and then did whole genome sequencing to look at the DNA copy number in the subclones. In the bulk, we see this average copy number between 0 and 1. When we look at individual cells, however, we see this very striking pattern that there's extensive variability between the different cells in their DNA copy number of the blue allele. In particular, we see there is some cell with small terminal loss, and that loss gets bigger in other cells in the same population. And we speculate that this varying pattern of terminal DNA loss could reflect some ongoing evolution in the progeny population. In other words, this could reflect some ongoing or continuous instability in the progeny population. Now, there are also, of course, cells with stable single copy state of the blue allele and cells that have lost all of the P arm that are also stable. But in between these two stable states, we see this spectrum of variation from very small terminal loss to a large amount of arm level loss. Now, when we turn to the red allele, as I mentioned earlier, there are segmental gains shown as the dots above the baseline copy number of 1. And then you can also see segmental interspersed loss on the left, where the large chromosomal segment that is having copy number 2 occasionally drops into copy number 1. When we look at individual subclones, each of them grown from a single cell, again, we see a lot of variability between these individual cells, often restricted to a particular region in the genome, highlighted here between the blue lines. In particular, you will recognize there are focal regions that are preserved in some cells but lost in other cells between 30 and 40 megabases. We went on to perform single cell profiling of the same population, and again, shown here is the P-arm copy number state from single cell copy number profiling that is consistent with what we see from the bulk data and also verifying what we see from the single cell subclone. Now on slide 30, I'm showing you another example that showed very similar feature as what we just saw in the previous sample for the blue allele. And in this case, we see that both the red homolog and the blue homolog shows this varying DNA copy number that is non-constant between different cells. And also that's highlighted with the thick sign line on the bottom, highlighting there's instead of a single sharp transition between different copy number states, it's a gradual slope between two different copy number states, reflecting this extensive variation in this bulk population of cells. So to summarize on this part, the outcome of copy number alterations generated by a single broken chromatid, what we have seen is in the first generation, the two daughter cells show this reciprocal terminal loss and gain. Now, the loss can persist into many generations, eventually terminate at the end of the centromere. This could lead to arm level loss. On the other side, the gain can lead to further segmental gains, including additional translocations, which would result in a stable or stabilize the unstable chromosome and leading to translocations both within the same chromosome as well as involving other chromosomes. What is intriguing is that in one of the samples I showed previously, there's also focal amplifications that start to develop in at least a subclonal population of cells. So what could contribute, or what mechanisms are generating the focal amplifications? Let's come back to this sample again. In this sample, we show that there are pieces or small segments between 30 and 40 megabases, the small segments of the red allele that seem to undergo dynamic evolution and development of a high-level amplification in some of the single cells. The ability to trace out chromosome rearrangements help us understand how exactly these fragments are generated. If you look at the very top population, the rearrangements we detected that join the different copy number segments are shown above the copy number plot. So here the black arcs represent joins between different segments. And the arc pointing upward reflects joins in one orientation, and arcs pointing downward reflects joins of the other orientation. I want to pay attention to this red arc on the right, which is also thicker than the black lines. When we zoom into this single translocation, what we see is that the translocation is not simply formed between the two ends as shown here. Instead, the translocation junction involves multiple tiny segments being hijacked across multiple spots across the genome. You can see this single event is being represented as this wiring pattern. If you follow the end from the far right, you walk the different curves. All of these curves jump back and forth, eventually land at the terminal point that was shown as the other end in the top. So this complex, we believe this complex rearrangement actually might play a role in generating the focal amplifications by putting all of these short fragments together. Indeed, as we look at the generational translocations from even the first generation, for most cases, we see the break of the chromosome or the chromatid was accompanied by a simple translocation. But also in a small fraction of cases, we see that the breakage of the chromatid was accompanied by very complex pattern of recombination events. Shown here, I've included 3 examples where the black curves and the magenta vertical lines represent translocations near the site of breakage that resulted from the initial formation of a dicentric chromosomal bridge. So we believe that the formation of complex chromosome rearrangements, including potentially the generation of many broken pieces of the chromatid resulting from chromosomal bridge resolution, contributes to the generation of focal amplification event. The exact mechanism still remains not completely clear, and we're working to resolve the detail of how this process could happen. One thing that I didn't have time to get into detail is we also observed that when multiple chromosomes were involved in chromosomal bridges, for example, when 3 chromosomes were both formed dicentric chromosomes and simultaneously broken, the break ends from the different chromosomes can join together to form this chain between different chromosomes. This pattern was sometimes known as chromoplexy, which is one different form of complex chromosomal rearrangement. To summarize on this part, we have talked about the wide spectrum of genomic alterations that can result from a single cell division abnormality known as the chromosomal bridge. Now, we know there are many other kinds of cell division errors, including multipolar mitosis, lagging chromosome, and you can see the chromosomal bridge of the bridge chromatin shown in green is shown on top, and formation of a micronucleus. Previously, we have shown that DNA of chromosome in the micronucleus can undergo this tremendous catastrophe, essentially resulting in a fragmentation of the chromatids and being distributed into 2 daughter cells. And this generates a mutational pattern known as chromothripsis, or fragmentation of a chromosome, that was frequently seen in the cancer genome. Now, the work I presented to you here showed that the generation of chromosomal bridges can potentially create something similar to a micronucleus, except that because the bridge chromatin was more localized to a segment in the chromosome, so it could result in this pulverization that's restricted to a more localized region in the chromosome. And we believe that the generation of abnormal nuclear structures, such as chromosomal bridges and micronuclei, might play an important role in generating the genetic alterations. Now, in the remaining of this talk, I want to share with you a slightly different story, which is on the other end of trying to understand genome evolution from in the context of tumor development. In this case, we try to study the evolution of genomic alterations in the process of, in the progression from Barrett esophagus, which is a known pre-malignant condition of esophageal adenocarcinoma, to esophageal cancer. This work is done in collaboration with Dr. Adam Bath's lab at Dana-Farber, with Matt Stetcher, who's not at UCSF, and a postdoctoral fellow in Adam's lab, Cheng-Zhong Zhang. So what we know about the formation of esophageal adenocarcinoma from Barrett's esophagus is that genome instability plays an important role in this process. This is shown here is a schematic diagram from a 2015 Nature Genetics paper from Adam Bath's lab, showing the 2 different pathways that a Barrett esophagus could progress to invasive adenocarcinoma. And in fact, there are 2 different pathways. One of them involves a peculiar step known as whole genome duplication when the cell fails cytokinesis. Another step doesn't involve whole genome duplication, but it involves loss of tumor suppressors, including P16 and TP53, that would essentially unleash a firestorm of genome evolution. Now, what we have done is we have looked at, we have harvested tissue from different stages in the course of progression from Barrett esophagus to esophageal adenocarcinoma, and profiled the copy number of these different lesions. So on slide 42, I'm showing you one example where we are comparing the copy number evolution, in this case of one particular chromosome, which is chromosome 18, in 5 different disease tissues, from Barrett's esophagus, low-grade dysplasia, high-grade dysplasia, to cancer. What you can see here is in the Barrett's esophagus, it is already obvious that, by the way, the figure here shown on the right are allelic DNA copy number. So what you can see here is even in the Barrett esophagus lesion, you can already see segmental DNA loss affecting a big chunk of the chromosome 18 arm. As we see, as we go down and look through the low-grade dysplasias, we start to see more events, including focal amplifications that arise next to terminal loss. This is seen both in the low-grade dysplasia as well as in high-grade dysplasia. Now, remarkably, some of these focal amplifications are shared, for example, between low-grade dysplasia 2 and high-grade dysplasia, but there are other focal amplifications that are private to individual lesions. For example, in low-grade dysplasia 1 and low-grade dysplasia 2, there are focal gains, in this case, even including known cancer genes, that are lost or not observed in high-grade dysplasia. Now, finally, when we go to esophageal adenocarcinoma, which is supposedly the most malignant disease, we actually don't see any sign of the altered chromosome at all. In fact, we infer that the chromosome that has undergone such dynamic evolution in the low-grade and high-grade dysplasias is completely lost in adenocarcinoma. On slide 43, I'm showing you another example. Again, this includes samples from the same patients with Barrett esophagus, low-grade dysplasia, high-grade dysplasia, and esophageal adenocarcinoma. In this case, I'm showing you the evolution on chromosome 17. We can recognize there is a very high-level focal amplification on chromosome 17 that's obvious from in both the low-grade dysplasia number 2 and esophageal adenocarcinoma. That includes both the ERB2 gene and the STAT3, as highlighted here. Now, when you look at the early stage lesions, including low-grade dysplasia 1 and high-grade dysplasia, you can already see some activity, some copy number alterations around the same region that is somehow shared between the low-grade dysplasia 1 and high-grade dysplasia, but distinct from low-grade dysplasia 2 and esophageal adenocarcinoma. Moreover, when you compare the low-grade dysplasia 2 and adenocarcinoma, you see very similar DNA copy number patterns, including extensive segmental copy number changes, both gain and loss on the terminus next to the region of focal amplification. So what this model suggests is that even though we generally think of high-grade or invasive disease as the sample with the most copy number alterations or with the highest level of genome instability, in fact, even in some of the early stage lesions, such events are already happening. And the reason that we have not seen this, perhaps, is because we often do bulk DNA sequencing, and sometimes when the harvest of the sample, when the sample we harvested is not pure enough, we're not going to be able to detect a subclonal population of cells that already have acquired such alterations. So to test this, we have performed a single cell DNA sequencing on one patient, a different patient from what was shown in the previous 2 slides. So in this patient, we profiled about 60 cells from a high-grade dysplasia that was biopsied by a pathologist. And we assayed their DNA copy number. So each cell is shown in a column, and each row represents a single chromosome arm. So from top to the bottom, it's chromosome 1 all the way through chromosome Y, and each column corresponds to a single cell. So to the left, you see a large population of cells that remain mostly diploid, except for occasional loss of chromosome Y, which is a frequent event in male patients. We then see cells with some level of alteration, most frequently on chromosome 17p, where the well-known tumor suppressor TP53 is located, and that is in the hypodiploid population. As we get to the right, we start to see more and more copy number alterations, and we inferred a population of cells which have become mostly tetraploid, which means all the DNA, each chromosome has doubled its copy number. And lastly, we see cells which has extensive arm level and whole chromosome copy number changes. They are mostly aneuploid, meaning they don't have a even chromosome counts for different chromosomes. In particular, in 2 aneuploid cells, we observed a focal amplification on the ERB2 locus that's next to a terminal, that's next to a large segmental amplification. In the same sample, we also see loss of heterozygosity on the 17p arm that was presumably related to chromosome, it was presumably related to TP53 inactivation. So what this data showed us is that the dynamic evolution of an unstable genome might have already occurred in a high-grade dysplasia, and some cells, after they have acquired hallmark events, in this case, the amplification of ERBB2 locus, would have the potential to develop into more malignant and invasive disease. In fact, the inactivation of tumor suppressors TP53 and P16 would enable the cell to tolerate this ongoing genome evolution and instability. And even if this cell doesn't evolve to become cancer, there's a good chance that some of the progeny populations descended from this cell would have the potential to do so. So with that, I want to conclude my presentation for today with a quick summary. In the first part of my presentation, I hope I've convinced you that even by generating a single broken chromosome, creating a single unstable chromosome can lead to a firestorm of genome evolution and mutagenesis. This can give rise to large terminal DNA loss, generate segmental gains, complex rearrangements, including the formation of focal amplifications. Although not shown here, we believe the generation of segmental and focal alterations is often related to abnormal nuclear structures, such as chromosomal bridges and micronuclei, which can potentially generate fragmentation of the chromatin in the structures. One, I think, profound implication of this result is that chromosomal instability, even though it started off with one single unstable chromosome, can lead to extensive genetic diversity and continuous evolution in the progeny population. In fact, even when we sample as few as 20 to 30 subclones, or 20 to 30 cells that develop into a subclonal population, they often show extensive variation in the DNA copy number. Finally, we think that the transition from precancerous lesions to cancer can be very dynamic, and genome instability might play an important role in generating diversity of genotypes that would provide a repertoire of cells that can be selected for under certain tissue context. Thank you for your attention.